Hi everyone, I'm Ling Chings and metric number is S71824 and presenting for molecular detections of viral nervous necrosis VNN in lattice calcariferous. This picture is showing lattice calcariferous and its common names is Asensi Barramundis, and giant sea perch. This species is tasty and nutritious meat that being loved by the consumers. So this species is known as fast growth, large size, and tolerance to wide range of environmental conditions, which it can live in freshwaters, brackish waters, and marine waters, depends on its life cycles. This make them to be first choice of farmers culture in the worldwide. This species has high demands in local and international markets, and the productions for lattice calcariferous in 2023, it produced 178,000 tons that values for 661 million USD. Now get some info for viral nervous necrosis (VNNs), or also can known as viral encephalopathies and retinopathies (VERs). This pathogen is under the genus of beta-nodovirus. This beta-nodovirus is non-enveloped, spherical in shape, and icosahedral symmetries in roughly 25 nanometers in diameter. This genome consists of two single strands positive sense RNA molecules, which are RNA ones with 3.1 kb and two RNA 2s with 1.4 kbs and without polyadenylates at the 3' ends. Here are showing the genotypes of VNNs. So in the early phylogenetics analysis of RNA to T4 variable regions, it been classified into four genotypes, which are SJNNV, triple jet nervous necrosis virus, TPNNV, tiger puffer nervous necrosis virus, Baffin's flounders nervous necrosis virus, BFNNVs, and the last is RGNNVs, red spotted grouper nervous necrosis. And for the TNNV is introduced in 2014. We've listed is the fifth genotypes for VNNs. This figure is showing the distributions of viral nervous necrosis VNN in the world. It's showing different genotypes found in different areas. And there are also two genotypes that I not mentioned before is re-exotens genotypes, such as RGNNVs, recombinants with SJNNVs, and another is SJNNVs combinations or with IgNNVs. In this table, it showing each genotypes has their own target host fish and optimum growth temperatures. For the SJNNVs, its serotype is A and target host fish is triple jacks. And the optimum growth temperature for this virus is 20 degrees Celsius to 25 degrees Celsius. And for the TPNNVs, its serotypes is B and target host fish is tiger puffer fish. And optimum growth temperature is in 20 degrees Celsius. For BFNMVs and RGNMVs is categorized in serotype C. So for BFNMVs, the target whole fish is cold water fish such as Atlantic halibuts, Atlantic cod, and flounders, and so on. The temperatures the optimum growth temperatures in between 15 degrees Celsius to 20 degrees Celsius. While for the IGNMs, is the target host is warm water fish, such as Asian sea bass, European sea bass, grouper, and so on. And the optimum growth temperatures for IGNMs is in between 25 degrees Celsius to 30 degrees Celsius. A report shows that has 120 cultural and wild species are infected in VNNs, that including fish and invertebrates. These VNNs infects all stages of fish and invertebrates, but mainly found in larvae and juveniles. This VNN's target organ is brain, spinal cord, and retinas. And the clinical signs for VNN infected fish is darkening skins, erratic swimming behaviors, such as wheelings, belly ups, swim rapidly in circles or straight heads, and lying down at the bottoms, tanks, and external and internal lesions like skin lesions, vein erosions, brain congestions, swim bladder hyperinflations, exophthalmia, and eyes damage. So there are two modes of transmissions for the VNNs, which are horizontal transmissions and vertical transmissions, where both transmissions will cause the healthy fish being infected. So for the vertical transmissions, that's transmitted through broodstock, gonads, and sperms, which the virus will spread to the next generations. According to some studies, that there are few species will spread VNN to next generations through vertical transmission, such as Asian sea bass, European sea bass, and striped jacks. And for the horizontal transmissions is through rearing equipment contacts with infected fish, feeds, and water environments. The diagnostic methods for detections VNN in fish and vertebrates are RT-PCR, nested PCR, and real-time PCR used. For the RT-PCR, reverse transcriptions polymerase chain reactions is the most rapid and convenient methods for detections VNN in clinically infected fish. So for the nested PCR and real-time PCR is using for detections asymptomatic fish. There are 5 different target regions for PCR amplifications with different combinations of primers. So for the target regions, once we produce the product of 1147 base pair with the combinations of F1 and R1 primers. For the target regions 2, we produce 875 base pairs with the combinations of F1 and R3 primers. And the third target regions is will produce 698 base pair with the combinations of F2 and R1 primers. And the target regions 4 is will produce 426 base pair with the combinations of F2 and R3 primers. And the last is the fifth target regions will produce 175 base pair with the combinations of F1s and R2 primers. Here is showing the roughly procedures for VNN detections using PCRs. The first step is sample collections from the brains or eyes tissues, and the second step is RNA extraction from the samples, and following by RT-PCR or the nested PCRs. And after the PCR procedures, we're going on for agarose gel electrophoresis and following by the RNA sequencing and phylogenetics analysis. Here are showing some VNN disease outbreaks in lattice calcarifer in the past 4 years. All cases of lattice calcarifer is categorized in RGNMV genotypes, findings that this disease outbreaks in two countries which are India and China, and all the fish showing the clinical signs such as floating on surface, hyperinflated swim bladders, abnormal swimming backwards, loss of appetite, skin pigmentations abnormalities, anorexia, brownies, and accompanied by body backlinings, and so on. Here are the sources that I'm using for preparing the slides, and that's all from me. Thank you.